to half of the adult US population and is the most commonly reported health reason for receiving soft tissue manipulation, commonly known as massage. STM is a form of mechanotherapy that imparts mechanical forces through the intact surface of the body and can be delivered by hand contact alone or instrument assisted with devices such as a rigid sphere or a flat blade. When performing STM assessments, the clinician simultaneously evaluates several tissue factors, including stiffness, mobility, and turgidity of inflammation. While the magnitude, angles, and timing of forces delivered to the tissue seek distinct clinical effects, STM practice mostly relies on subjective, nonspecific, and qualitative descriptors. Progress to show the direct effects of soft tissue therapies on patient outcomes will require the development of quantitative biomarkers with robust signal-to-noise ratio among patients. Numerous methods have sought to characterize the viscoelastic properties of biological tissue. For instance, ultrasound-based methods measure displacement, shear wave differences, and microvascular changes to perturbations in muscle and nerve tissue. Myotonometer devices use accelerometer recordings to assess the tension, elasticity, and stiffness, and other imaging approaches such as optical coherence tomography capture small planes of tissue to estimate tissue layer thicknesses and relative density. While these methods can measure the mechanical properties of tissues, each is limited to localized regions of interest, usually spanning less than 10 square millimeters, and thus must be sequentially repositioned in order to characterize larger muscles. In contrast, we developed an approach to optically measure a larger field of deformation at the skin surface simultaneous to soft tissue manipulation. This video shows a licensed clinician performing STM assessment on the cervicothoracic or upper back region of a patient. The blue rectangular patterns of the patient are used for post-analysis using a 3D DIC method, which I will discuss in a few slides. An added benefit of this method is that it does not impede the physical touch between the clinician and the patient. In the study shown on the left, a thin film of tegaderm decreases recipient pleasantness during an affective touch brushing study. Similarly, the work on the right shows a decrease in mechanical deformation as measured by strain when the index finger is covered with a latex glove. These works validate prior studies that show physical barriers on the skin, as well as instrumented touch, have diminished C-tactile response as compared to human-to-human contact, and in effect, attenuate pleasantness and tactile perception. In this work, we use a surface imaging approach to capture large areas of the participant's upper back region during STM assessment performed by a licensed clinician. From the 3D displacement data, we developed a skin mobility biomarker based on surface stretch during the manual pull mechanism of an STM assessment. Case study results show that the skin surface biomarker differentiates bilateral stiffness within a participant, with trend directions agreeing with those of the instrumented force probe. Lastly, this method does not interfere with the psychosocial benefits of human-to-human touch. To determine a baseline measurement for tissue stiffness, an instrumented force probe is used to capture the force displacement response of a rigid sphere upon its manual indentation into the skin and myofascial tissue of a human participant. The normal force is collected by a 6-axis load cell in the instrumented probe, and the displacement is tracked by DIC imaging. In the presented plot, we can see that the points in green on the participant's left side reach forces of approximately 15 newtons at a displacement of approximately 5 millimeters. From the data, the participant's left side, in green, can be characterized as more stiff as it reaches similar levels of force at lower levels of displacement. In this work, we use a non-contact optical tracking method called 3D digital image correlation. The method works by tracking unique pixel patterns across image frames and uses a camera calibration step in order to transform 2D pixel tracking to 3D deformation data. To do so, it requires a speckle pattern to be applied to the surface of interest, as seen above. The density and randomness of points in the speckle pattern inform the resolution of the 3D data structure. I've used this method previously to characterize the skin states associated with von Frey monofilaments. From the raw image of the speckle pattern deforming when indented with a monofilament, we can track point-specific displacement, resulting in 3-dimensional strain fields. In this work, at the beginning of each trial, participants had a speckle pattern applied on the upper back bilaterally via an ink-based stencil transfer paper method. They then lay on their stomachs on a portable massage table, above which 3 cameras were positioned to collect imaging data for the DIC pipeline. In these experiments, 2 types of soft tissue manipulation, manual compression and manual pull, were performed to evaluate the myofascial tissue of the cervicothoracic region. For the scope of this talk, I'm going to focus only on the manual pull mechanism. In manual pull, static force is manually applied in 4 lateral directions: superior towards the neck, inferior away from the neck, medial towards the midline, and lateral away from the midline, from a central focal point, usually a tender spot. Clinicians use this technique to assess the distance and velocity of force application and the point at which discomfort, if any, is reached. Here we can see measurements of displacement and first principle Lagrangian strain as calculated by PVDIC during manual pull. The left panel shows the clinician applying a pull in the medial direction, and in the right panel in the superior direction. You can see that the maximum displacement and strain occur at the point of contact and diminish outwards away from contact. To develop the skin mobility biomarker, we first look at the 3D displacement of the surface over time. From the 3D vector plot above, we can identify the direction of maximum displacement over the entire surface, which informs the direction of manual pull by the clinician. Applying this classification to each image frame, we can characterize the 4 pull directions over time. Next, we examine the first principle stretch, which quantifies the change in distance between tracked pixel points of the 3D point cloud. A stretch value of 0.8 depicts a 20% compression between tracked points, while a value of 1 depicts no change. We can see a lower stretch value or a larger compression is observed near the point of manual pull, as indicated by that dark blue color. At each time point, we measure the minimum first principle stretch over the entire point cloud. After plotting these minimum stretch values over the time course of each trial, we can see 4 distinct valleys that align with each applied pull. We then take the minimum stretch value for each pull to compare participants' left and right anatomy. As seen in the plot above, a clear separation in minimum stretch is found over 4 directions between the left and right side for this participant, with the left side green showing lower levels of stretch and as such more compression. We use the DIC method to measure the magnitude of pull delivered in each of the 4 directions. The skin mobility biomarker is then defined as the relationship between the minimum surface stretch and the maximum pull for each of those 4 directions. This data is from 1 participant over the course of 2 trials and illustrates clear bilateral separations in the superior and medial directions. As discussed previously, baseline stiffness measurements are made by an instrumented force probe. For the same participant I discussed earlier, we found their left anatomy to be more stiff than their right anatomy. Looking now at the skin mobility biomarker, we observe clear bilateral separation in that superior direction. In specific, at a maximum pull of approximately 2 millimeters, minimum skin stretch is measured to be 0.56 or 44% compression on the left, while only 0.93 or 7% compression on the right. Similar left and right distinctions are observed for 2 more participants, with the latter participant exhibiting higher ratios of compression to pull on the right side of the body. In contrast, the last participant exhibited overlapping bilateral regions as measured both by the instrumented probe and the skin biomarker, suggesting no bilateral distinction. As seen above, more skin stretch at similar levels of manual pull is found in tissue that is characterized as more stiff. We believe that when the myofascial tissue is stiffer, it's more resistant to change, and thus the skin must stretch more to account for that applied pull. On the other hand, when the underlying tissue is less stiff, it is less resistant to change and moves more fluidly when the skin is pulled, requiring less skin stretch. In summary, this work used a surface imaging approach to capture large areas of the participant's upper back region during STM assessment. From the 3D displacement data, we developed a surface biomarker and found that it was able to differentiate bilateral skin mobility with trend directions similar to those from an instrumented force probe. Furthermore, this method allows for the psychosocial benefits of human-to-human touch between clinician and patient. Since this study, I've collected a new data set with an increased sample size, as well as begun the first steps towards a longitudinal exploration through the use of a semi-permanent speckling method. In future work, we would also like to examine the contextual factors of clinician-patient relations during STM, namely how individual differences may influence the effect of treatment. This work is part of a group called Neurons Matter. Neurons Matter is an NIH-funded network to further our understanding of how massage and other mechanotherapies relieve pain and promote tissue healing. It's a wonderfully inclusive community, and if you'd like to join, please scan the QR code above. Thank you to my lab mate, Zach, our clinical collaborator, Terry, and my advisor, Greg. Thank you for your attention. I'd be happy to answer any questions.